My name is Xu Hakubo Zhu, and I'm from Weda Kitakuchi lab in the laboratory for chemistry and life science. And today I'd like to introduce my research at Tokyo Tech with the title Fluorescent Immunosensor Meets Ultra-High-Throughput Screening. And I will first introduce what is the fluorescent immunosensor. So when you hear about this sensor, you may think it's a kind of small device and that may can detect some targets. But in this case, the fluorescent immunosensor, it means protein, a small protein, and we put this protein in the small test tube and mix with your target, and you can read the fluorescent signal using a fluorometer. That is very simple and relatively low-cost method to perform the measurement of a huge variety of different targets. And since this type of immunosensor was made from the antibodies, and so that means the targets almost is infinite. And so and here you can see this detector is kind of small, so it's really have a promising application for the remote diagnostic or the on-site detection of your targets. And in this slide, I will introduce which type of immunosensor we're using now. The name is called Quench body, and Quench body is derived from the antibody fragment shown here from the here's the IgG from the human body, and you could see from this structure here. So we take part of it and labeled with fluorescent dye. And when it meets the target, the fluorescent fluorophore will be exposed and then can be excited by the laser or different LED detectors. And we can rate the fluorescent signals. And we just simplify this like the light bubble and bulb and mix with target, and it lights up. Just a simple image of that. And here is the example of detecting the SARS-CoV-2 spike protein using the QBody we developed recently. So in this image, you can see we just mix the saliva sample from the human and the spike proteins mixed in the test tube with or develop the QBody biosensor. And when increasing the spike protein concentration, and the fluorescent signal successfully increased, and we can rate the fluorescent signal and correlate it to the spike protein in the saliva to show if the patient is infected or not. And this is quite a simple demonstration. And the good point is we can finish this measurement within one minute. It's really fast and to do the quick screening for the coronavirus. And after we see this example, we want to show what is the challenge part when using this type of the QBody immunosensor. Actually, you can see different parts on the sensor always need to be optimized. For example, which fluorescence dye is best for this sensor? And what is the optimal linker length and sequence here? And where the additional quenching motif should be induced inside the antibody? Or do we need to improve the binding affinity, and how we could improve that? And do we need to improve the stability and product yield? And after we do the optimization, will the sensor still functional? So this is all the challenges. And the final thing is, what is the best combinations? There's so many factors. When we do the engineering, usually we need to take the one person to do the engineering continuously for several months to a year. That's really time consuming. And since there's not that systematic or comprehensive optimization, it's hard to make the solid conclusion and which can be used for the next sensor development. So what can we do? Can we use the ultra-high throughput screening method to screen all the possibility of these combinations and find the best one. So this slide shows the previous research I performed. And in this research, I used messenger RNA display technology. This technology can handling trillions of different variants, and combined with the next generation sequencing, we can get the millions of the selected sequences, which will provide a lot of information to make solid conclusion. So briefly, we have the DNA library encoding the protein or peptide, and we do the transcription translation and get this messenger RNA and the peptide or protein conjugate. And then we stabilize it with the cDNA and immobilize it on the surface with a certain method. And we test the function and do the sequencing and further analysis and combine all the information to get the information we need. So when it comes to the QBody, what can we do? So similar thing. So we have the DNA library, like here, and transcription, get messenger RNA, and do the pyrimidine ligation, translation, reverse transcription, and then we will have the cDNA antibody library. This thing is already exists, but what if we label this antibody with fluorophore? Will it be still functional on the cDNA? And can we select the best QBody from this cDNA library? That is the most challenging part in this research. And actually, I could not show the data already collected recently, but I have already know the display of the QBody on phage is possible, and we could also select the good variants from the displayed library. So that will be a kind of a proof indicate this type of the high throughput screening method will be suitable for QBody development and information collection. So I will really expect to get a kind of more solid result to share with you, but currently I could not. But in future, I will have the chance to share my result. And thank you very much for your attention. And this will be all the contents of my presentation today. Thank you.